Hey guys, it's Izzy. So today is the fourth lab for my Forensics 106 course at my college. I lost track of time A for my first one. B, I got really sick around the crime lab one. Where it was like a crime scene where they gave us like a map and we had to like answer these questions about it. And then last week I still like was not feeling good from my stomach bug, so I ended up just like rushing through it all in like a couple days because I had like training from work and like a bunch of other stuff. Anyways, so today it's a DNA extraction lab and I'm gonna film the whole process, but this is my DNA I took out. Yay!
We want to add about 4 milliliters of water to this. If you suck the water up into the transfer pipette, this top line here, that's exactly 1 milliliter. We don't have to have exactly 4. We're going to have around 4 milliliters of water. So I'm going to suck up about 4 of these pipettes worth of liquid. It's going to fill the test tube up about a quarter of the way.
Wait, which one are we filling up?
Test tube is 4 milliliters. Suck the water on liters on it. And we want to add our transfer. We need to mix up a soap solution. So what we're going to want to do is we're going to take one. All right, so we need to mix up a soap solution. So see that on the experiment be first mix up a soap solution. So what we're going to want to do is we're going to take one of our transfer pipettes, one of the graduated ones that has the markings on it. We want to add about 4 milliliters of water to this. If you suck the water up into the transfer pipette, this top line here, that's exactly 1 milliliter. We don't have to have exactly 4. We're going to have around 4 milliliters of water. So I'm going to suck up about 4 of these pipettes worth of liquid. It's going to fill the test tube up about a quarter of the way.
Oh, that's not a quarter. Oh, his is probably bigger, huh? Why is in the wrong one?
Once I've added my 4 milliliters of water, I'm now going to add just about 1 drop of this liquid dish detergent. I don't want to add a lot. I'm just going to add about 1, maybe 2 drops.
Drop.
That's okay. Now what I want to do is mix it. So I'm going to use the same transfer pipette that I used to add the water. And I'm just going to go ahead and mix it together. I can do that simply by squeezing the transfer pipette several times. The other way to mix it is to simply just tap the bottom here. So now I have a solution that's part water, part soap. Right, the next solution we want to mix up is the one labeled enzyme. And so
Pause.
with the enzyme solution, I'm going to fill this up again about 4 milliliters. So I'm going to fill it up about 1/3 of the way full.
It says 130, but this is bigger than mine, I'm pretty sure. Unless I'm using the wrong ones.
Again, this is going to be somewhere between about 1/4 to 1/3 of the way full. Not really exactly, but look for now what we're going to add is we're going to add a little bit of meat tenderizer. Now I did want to make mention, you notice that for this particular experiment, I'm not wearing any gloves, I'm not wearing a lab coat. And that's because all the things that we're using for this particular experiment, none of them are hazardous or caustic or toxic. And so meat tenderizer, we use this all the time on steaks or hamburger. And so we're just going to add just a small amount of meat tenderizer to our test tube. We'll go ahead and unscrew this here. We're going to add what equates to about 1 pinch. So if you were to just take your fingers and pinch the drop, and then you could pour out next to it and use a small one. So I'm just going to about 1/16 of the time.
Does this what his look like? No. I might dilute it. Okay, and then we go for the soap, he said, which or salt, soap.
Granules are dissolved in our liquid, so that mixes up our enzyme solution. Right, the other solution that we need to mix up is simply a saline or a salt solution. So get this particular test one, about 4 liters of water. So you're going to fill that one first. Really full. Next to this test tube, I'm just going to add about 1/8 teaspoon of total salt. So add salt here. So we know this is salt solution at the bottom of the test tube. So we have a saturated solution salt. I'm adding a little water, so we have a little more salt actually. Solution. Maybe 3, maybe 4 more. So there versus 10. So we have a salt solution, we have an enzyme solution, and then we have our soapy solution. And then of course we mixed up our solutions, and now we're ready to actually do our extractions. So one of the solutions we mixed up was a solution that was a mixture of water and a little bit of dish soap. So what we're going to do now is we're going to take our test tube that we previously labeled as cells, and we're going to add about 1 to 2 milliliters of our soapy solution to this test tube. So add 1 or 2 milliliters. In fact, maybe even 3.
We're gonna put 1 because I don't think this is enough.
Now, so what we have now, this is just our soapy solution, but we need to put some cells in there. So what you're going to do next are about 3 or 4 clean cotton swabs. These are just cotton swabs or Q-tips that you might use in your bathroom, and I've grabbed these. What we're going to do is we're going to collect cells from the inside lining of our cheeks, or what we call buccal swabs. And so I'm going to take these cotton swabs and I'm just going to rub them on the inside. Our clothing is soaps are very good at dissolving fats, oils, and things that get on our clothing or get on our hands or on our dishes. And so as we put the cells inside this test tube, that soap is slowly dissolving the cell membrane on the outside of the cell, and then also dissolving the nuclear membrane that surrounds the nucleus so that the DNA can then separate out and then dissolve within our liquid. So that's what we're going to do first. We're going to go ahead and put our cells into the solution that contains the soap and the water. All right, so that's step 1. The next step in our extraction process is to add to our test tube that contains our cells dissolved in the soapy water solution. What we're going to add to that test tube is a few drops of this enzyme solution that we mixed up earlier. Remember, this enzyme solution is basically a small amount of meat tenderizer that has been dissolved in some water. And to add the meat tenderizer or this enzyme, I'm going to use one of the micro.
Cells are here. 1, 2, 3, 4, 5, 6, 8. I think I marked these better. Ow!
Meat tenderizer. Now, the meat tenderizer consists of what's called a protease enzyme, which is a protein dissolving enzyme. So we just add the protease enzyme to our solution. So we added that to our solution. We mixed it by tapping the bottom of the test tube to mix it in there. And then we needed to let it sit for about 2 to 4 minutes. And so after we add the enzyme and we mixed it, and we let it sit for 2 to 4 minutes, we're ready to do our next. Our next step is going to involve again one of these micro pipettes or these pipettes with the micro tip. What we're going to do is we're going to take our salt solution, which we mixed up earlier, and then we're going to add about 8 drops of this saline or salt solution to our mixture. So I'm going to add about 8 drops test tube and hold it at about a 45-degree angle.
Okay, and this is when we hope it's frozen enough.
Close. You can actually see there we have
Bye.
a 2-layer liquid. You can see that there's 2 separate layers. And the alcohol layer is there on the top. And then in the bottom layer, that's where we have our soapy solution with our DNA dissolved. I'm going to go ahead and set this down on my test tube rack. Be very careful and put the, because I don't want to mix them. I'm going to put my test tube down. I'm going to let it sit for about 4 or 5 minutes. I don't want to disturb it. I don't want to hit the table or mix it. Precipitate out of solution, and so we can actually see our 2-layer liquid. And normally the DNA, most of it actually is going to sit right between the 2 layers, but we actually have a fairly large piece of DNA that's kind of floating in the bottom layer. It's kind of hard to see on the camera. I'm kind of hoping if I hold it up to the light here that we're going to be able to see it a little bit better. If you look there, you can see a kind of a stringy substance kind of suspended in the test tube. If we look at the bottom of the test tube, if I can get the camera to focus in a little better here, most of our DNA actually has settled down to the bottom. So now what we need to do is we actually need to suck that DNA out of solution and actually deposit it into our micro pipette, or sorry, not a micro pipette, but our micro test tube. So I'm going to hand the camera off to my cameraman. I'm going to show you how to go ahead and do that. Alright, so our DNA, after we added our alcohol and let it sit for 4 or 5 minutes, is now precipitated out. So we get little teeny strands of DNA that are sitting right between the 2 layers, and then a lot of it actually has settled down to the bottom. And so now we need to suck that DNA out and transfer it to one of our micro test tubes here. So I'm going to go ahead and have my cameraman kind of zoom in on the test tube a little bit. And the way you're going to do this is you're going to take the transfer pipette, and the first thing you want to do before you put it in is you're actually going to squeeze it just a little bit. Just actually squeeze it first. We don't want to dip it in there and squeeze it, because then we'll actually make air bubbles. We don't want any air bubbles. We actually want to squeeze it first, and then when we let go of it, it's going to suck up the materials we want. So we're going to go ahead and place our pipette in here. We squeezed it a little bit before we put it in the liquid. We're going to put it down, the tip of it down to the level where we need it to be, where our DNA is. And then we're going to suck up kind of that little bit of that middle layer. And I'm going to transfer that liquid to my micro test tube. And then a lot of the DNA was found at the bottom of the test tube, so I'm going to, again, grab my large test tube, and again, I'm going to try to suck up that small amount, that very, 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 very bottom section that had a lot of our DNA that kind of settled down to the bottom. And I'm going to go ahead and put that into my micro test tube. So now I've taken my DNA out of here, and I've now transferred it into this much smaller micro test tube, and I go ahead and cap it. And so now I have my DNA that's been extracted. So in the crime lab, our next step would be, after we've extracted the DNA, would to be to amplify it, which is to actually copy it, so that then we could develop a DNA profile from the DNA that we have extracted. So when you're all said and done at the end of the exercise, this is what you're going to get. You're going to have your DNA extracted and collected and concentrated in the bottom of your micro test tube. So that's the DNA extraction exercise. I hope you guys enjoy it. You can actually keep your extracted DNA, and then you can make a clone of yourself at some future point if you want. So anyway, that's the extraction exercise.
Yeah, I think this was a lot of fun. Thanks, Professor.